Before we move on to look at proteins, let's take a quick review of what we've learned about nucleic acids. First of all, there are three things that we need to remember about every one of our polymer macromolecules. The first is, what is the polymer? The second is, what's the name of the monomer? And the third is, what is the bond name? They're all covalent bonds, but they have specific names depending on the molecule involved. Here we were considering nucleic acids. So the polymer of a nucleic acid is indeed a nucleic acid. What is the monomer of a nucleic acid? Monomers were nucleotides. And the nucleotides are held together by phosphodiester bonds. So the nucleotides are composed of three subunits. Do you remember what those subunits were called? One was a pentose sugar. Off the 1' carbon, there was a nitrogenous base, and a phosphate off the 5' carbon, as well as an OH group off the 3' carbon. That brings us into three N types of words that we need to consider. We've got nucleotides and nucleic acids and nitrogenous bases. So let's draw them out somewhat schematically. These can become slightly confusing for people. The entire macromolecule class is called nucleic acids. And nucleic acids are built up of nucleotides. And one of the components of the nucleotide is a nitrogenous base. Keeping these three words that begin with N separate and understanding their magnitude is going to be very important for your understanding of nucleic acids. But now let's move on to examine proteins. Again, you'll need to keep track of the polymer, the monomer, and the name of the bond. Proteins have a lot more diverse function than any of the other macromolecules. They can either have structural roles or functional roles. That is, they can either do things or they are things. For example, enzymes are proteins that do things. They convert one molecule into another molecule. We'll also see that proteins are involved in defense. These are also doing proteins. So things like our immunoglobulins and antibodies are involved in defense of our body. Proteins can also act in transport. They help transport substances across the cell membrane. Then we'll have some structural proteins, ones involved in support, like collagen fibers and keratin fibers that give our cells some structure. Proteins can also be involved in motion. For example, muscle is full of actin and myosin filaments that are proteins that allow us to move. Proteins also have a role in regulation, as well as in storage. Proteins are polymers that are composed of one or more long, unbranched chain of amino acids. Each chain is considered a polypeptide. We might represent amino acids in a chain forming a polypeptide, much like this. Just a string of beads. Each one of the beads is an amino acid, and they are strung together by peptide bonds. So there we have it. The polymer is a polypeptide. The monomer is an amino acid, and the bond is a peptide bond. Let's look in a little bit more detail at amino acid structure. An amino acid has a central carbon. You can see that exhibited here. On the central carbon, there are three parts that will always be there. First of all is the amino group, this H2N located over here on the left. Second is the carboxyl group, or COOH. And then the third will be this single hydrogen. The R group here is the group that is interchangeable. R could represent many different things. It's the variable group of the amino acid. You should be able to draw an amino acid just as simply as this. We can draw the central carbon. We can have an H group. We can have our carboxyl group. We can have our amino group, and then the R group. So go ahead and diagram an amino acid for yourself now. So the R groups can come in several different forms. They can either be in our nonpolar form, or polar uncharged form, or actually a full-on charged form. You can see there's a great variety of R groups on each of the different amino acids represented. On the amino acid alanine, it's a very small, simple side group. Here's another side group that we see on valine, and an even bigger side group that we might see on isoleucine. Again, they can be nonpolar, they can be polar but uncharged, or they can actually be charged. Now, remember water. Water is hydrophilic, right? Water loving. Things that are nonpolar, were they considered to be hydrophilic or hydrophobic? Remember the example of sodium chloride and dissolved in water, where the polar ends of the water molecules were attracted to other polar things. So water is polar, and things that are polar like to be around water, whereas things that are not polar are more like oils and don't like to be around water. So when we put any of these amino acids in solution, all of the polar portions of molecules will like to stick together. So for example, this side group is a polar side group, and so it would be attracted to molecules with other polar groups. When the molecules have charged side chains, we have a similar situation where negatives are attracted to positives. So amino acids, just like all other monomers in polymer macromolecules, are joined to each other by dehydration synthesis. Here, an OH and an H are snapped out in order to produce water. We've dehydrated the molecule by pulling water out of it. Now we have a dipeptide. In order to form a polypeptide, we simply do the same thing, stringing these together end to end. Polypeptide chains fold in various ways to form their three-dimensional shape, and the shape of the protein really determines its function. Here's an example of some attractions between various regions of molecules that might cause the polypeptide chain to fold and bend into various shapes. In here, in the green area, you can see that this is a hydrophobic region. Let's say this protein was floating around in a cell. It's in an aqueous environment or a hydrophilic environment. So anything that's hydrophobic, that is the non-polar portions, will naturally fold to the inside in order to stay away from all of the water molecules. So let's take a closer look at these different ways that proteins fold. There are 4 levels of protein structure. The first level is the primary structure. That's simply the sequence of amino acids, or the beads on the string that we might draw like this, simply representing an amino acid as a bead, connecting it to the next one, and the next one, and so on and so forth. So we could have any one of the 20 amino acids here. This one could be alanine, this one could be tryptophan, this one could be phenylalanine, this one could be arginine, so on and so forth down the string. Here are the beads represented. So each one of these boxes represents one of the bead joined together by a peptide bond. Here we're representing a peptide bond. It's simply a covalent bond. So the primary structure, simply the order of amino acids in the polypeptide chain. Next, we look at the secondary level of structure. This is where we have interaction of groups of the peptide backbone. The most common secondary structure formations that we see are the alpha helix, in which the string of amino acids, or the primary structure, forms loops, just like a DNA helix. The other is the beta pleated sheet, where we just see kinks along the length, one amino acid, another amino acid, creating these kinks or pleated sheets. The pleated sheets usually come in sets of 2, so that bonds form between amino acids in a different pleat. So the primary structure, simply the beads on the string. The secondary structure, the formation of alpha helices and beta pleated sheets. In tertiary structure, this is the final folded shape of a single globular protein. It's stabilized by a number of different forces. And tertiary structure involves only one polypeptide chain still. It's the final level of structure with proteins that consist of only a single polypeptide chain. You can see a number of different alpha helices and series of beta pleated sheets, all on one single polypeptide chain, many beads on the string. The final level of protein structure is the quaternary structure. This is where we see many different polypeptide chains coming together, 2 or more. They're individual chains or subunits of a protein. Each have primary, secondary, and tertiary structure. Hemoglobin is a perfect example when we see the coming together of 4 separate heme groups in order to create the whole hemoglobin molecule, which is composed of 4 separate polypeptide chains, each with their own primary, secondary, and tertiary structure. So quaternary structure is when we have many subunits or many polypeptides that have assumed tertiary structure coming together to form one globular protein. So here's a review of each of the layers of protein structure. In your assignment for this chapter, you'll be asked to diagram the 4 levels of structure of a protein. Here's a great place to use for some inspiration for that diagram. Now, there are some additional structural characteristics that researchers have noticed over the recent years. There are motifs and domains. Motifs are common elements of secondary structure that are seen in many polypeptides. Things like a helix, turn, and then helix comes up very commonly. Or a beta-pleated sheet, an alpha helix, and another beta-pleated sheet. These motifs come up very frequently in all sorts of different proteins. One might make the analogy that if the amino acids are the letters in the language of proteins, then the motifs represent words that are repeated throughout the language continually. Domains are much larger than motifs. If we carry out that analogy a little further, the domains are like paragraphs that might be repeated over and over. They're functional units with much larger structure. Most proteins are made up of multiple domains that perform different parts of a protein's function. So domains, again, are paragraphs that are common to the language of proteins, whereas motifs are like words that come up over and over. So domains are much larger repeated patterns in the folding of proteins. So it was once thought that newly made proteins folded into these structures spontaneously. But researchers have recently discovered these proteins, which we call chaperone proteins. They help the protein fold correctly. If we waited for the hydrophobic forces and hydrophilic forces to pressure the protein into shape, it would literally take forever. It's noted that deficiencies in these chaperone proteins are implicated in certain diseases. For example, cystic fibrosis is a hereditary disorder. In some individuals, the protein appears to have the correct amino acid sequence, but it fails to fold properly. This is an example of a chaperone protein. Basically, the misfolded protein or the unfolded protein will go into this capsule. The capsule does something to it in order to help the protein refold into its original shape. And it's released as a correctly folded protein. It's not fully understood exactly how these chaperone proteins work. So high temperatures cause proteins to unfold or become denatured. And there's a class of chaperones called heat shock proteins that help refold these proteins that have been shocked by the heat. So when a protein loses its correct structure, it also is unable to perform its natural function. Denaturation can occur for a number of different reasons. Temperature is one of them. But a change in pH can also alter the structure of a protein significantly. Or if we change the ionic concentration of a solution in which a protein resides, we'll also see that it begins to unfold. You've all seen denaturation when you fry an egg, where the white becomes firm. We can often never return from that denatured state. So as you can tell, it's very important that we're able to buffer pH and maintain temperature in homeostasis, as well as the ionic concentrations in each of our cells. This slide is a reminder that there are many animations for you to view on the Connect website. If you're having trouble understanding any of these concepts, the animations will be really, really helpful in helping you understand. So please take a look at those animations before you move on to the next section.